Okay, you guys ready to get started? This is the last week. Can you believe we've made it? Almost. You guys are going to be super happy that you're here today because we're going to play some Jeopardy games to help you study in the second half of class, and you're going to get a lot of extra credit for them. So you'll be super happy that you're here today. It should be the 12th. Remember how I handed out that syllabus adjustment because it turned out that all of the dates were wrong. So I handed out a paper copy of the syllabus adjustment. So it's Thursday. PIMAL is due by next Monday. Is that what? No, I'll give you till Monday. It's not a big deal. So, yeah, so just by 11:59 p.m. next Monday. If you have trouble uploading it on Tegrity, just send it in an email to Ben and I, okay? Because I think Brad had some trouble uploading his, so if you have trouble, just send it. And that's not a big deal. We're basically going to be incommunicado starting on Friday, well, on Saturday. So we won't really be necessarily around to help, just because we're going to be driving to Alaska. So we may have signal, we may not have signal. If we do, we'll answer. So for the rest of the week, it really is more, I've got like 7 slides on the regulation of gene expression to kind of hit some of the highlights that may be on the final that I don't feel like we covered well enough when we talked about genes. And then we're going to go through and essentially just play some Jeopardy games and try to refresh your memory on some of the earlier stuff that we did. Okay? And I think Ben, he's going to lecture on the immune system tomorrow. Okay? So it's a great question. Because they're going to be Scantron, paper Scantron sheets, make sure you bring pencils. It depends on how quickly I can get them scanned. I'm hoping I can get them scanned and uploaded on Friday. Otherwise, I'll probably leave them with my technician, and so she can do it on Monday. But it means that if I have internet access, I'll finalize your grades while I'm in Alaska. If I don't have great internet access, you're going to have to wait, and you'll just have an idea of how long. You won't have a finalized grade yet, but you'll at least know how you did on the final. Okay? I'm hoping Friday, Monday time frame. But then because the PIMAL videos, we're going to have to look through those. That's why I can't tell you you'll have your finalized grade. Yeah, so you kind of know where you are. That's the plan. Okay? You also have to keep in mind that, remember, the final test is not, it's worth like 300 points for our class, but it's not that many questions, so it'll scale, and it will probably, well, it will likely be curved. So you're going to see that you got like 17 or you got 20. Don't freak out on me because there's going to be a lot of adjustment that goes into that. Okay? So there will be a lot of adjustment that goes into that. Okay, so when we get to the part where we're going to play the game and you're going to earn extra credit, we're going to divide into 2 teams. You earn the amount of extra credit that your team earns, but we rotate through who's answering the questions, and you can work as a group. You can look things up. I don't really care. And you're keeping track of your own points, and then at the end of class, you'll hand me your points so then I can put those in the extra credit column. Sound good? Great. Okay, so we're just going to finish up with a couple of things here on gene regulation, because we've talked a lot about transcription and translation, and we've kind of mentioned how genes are regulated, but I think it's important to explicitly talk about how genes are regulated and their expression. So your objectives today for this part is to define general principles of gene regulation and identify differences in the regulation between bacteria and eukaryotes. So that's really what I want you to get out of today. So there are 7 general processes that can affect the way genes are regulated, and they're listed here. So you have transcription right up here. You have post-transcriptional processing. So this is going to be like your methylation. It's going to be some of your phosphorylation that you end up getting on the DNA itself. And sometimes people throw in some of the splicing variants in this post-transcriptional modification. And then remember, we can have mRNA degradation, so how stable is that RNA going to be? That can also be a form of regulation. Ooh, it just did all sorts of things here. And then we get into translation, and that gives us our protein level. So again, we have our post-translational processing. And so that's going to be like your ubiquitination, which, remember, that's that signal that would lead those proteins to be degraded. It can also be your phosphorylation, your methylation type things like that you're going to get on your proteins, not on your DNA. And then you can have your protein degradation. So we can actually, again, tag it for degradation or make it degrade at different rates. And then the final step that we can regulate is protein targeting and transport. So how are these things going to move out of the cell and actually do their work? So again, we can either have negative regulation or positive regulation. So we've had our 7 steps, and now we're going to divide it up a little bit further. And again, we have 2 forms of negative regulation and 2 forms of positive regulation. Now, eukaryotes mostly use positive regulation. So essentially the difference is between 1 and 2. Here you have your signal molecule here, and then you have the promoter can sit right here on the operator, and then your signal molecule can bind to the promoter down here, versus over here where you have the molecular signal is what causes the binding to the regulator or to the operator right here. Now that's different than our positive regulation, because in our positive regulation we can have, again, it's just the order in which things bind. So we can actually have our signal molecule bind with our promoter versus it binding when it's already on. And so it's just an order event. And you don't really need to understand necessarily the order. You just need to understand that there's both positive and negative regulators. So then there's this kind of interesting thing called an architectural regulator. And so here we're actually changing the shape of the molecule. This is mostly going to be in your eukaryotes. It's not as much done in bacterial cells because you have these very complex interactions between a lot of different subunits of proteins. But you can see here that we have a DNA bending protein that comes and sits right here, and it actually changes the shape of the DNA to allow and facilitate the interaction of all these different elements. So the next thing that bacteria do that are very different than what eukaryotes do is this thing called operons. Have you guys ever heard of operons? A little bit, probably. So remember that our operon is basically, you've got a bunch of genes that are all under the control of a single promoter in an operon. This is very different from the way eukaryotes do things, where they have single promoter, single gene. Here you have multiple genes that are all under a single promoter. And kind of the classic case that I'm showing you here is the lac operon. where lactose releases the repression. So you have your repressor that binds to your operator and prevents RNA polymerase from moving and working on these genes down here. But in the presence of lactose, it binds to the repressor and releases that repression. And so this is how your LAC operon works. This is a big part of how we produce recombinant proteins in bacteria. So a lot of it is based on this LAC operon. So there are a couple different domains that we need to talk about. So we have our helix-turn-helix domain that's shown up here. This is a simple structure regulatory protein. It's common in prokaryotes. It binds to the major groove of DNA. So you have your DNA binding helix here, and then you have a turn, and then you have another helical region here. There are, again, approximately 60 to 90 amino acids. You have your zinc finger domain, where you've got this coordinated zinc ion that's kind of right at the base, and then you have a recognition element here that recognizes the helix. So we're talking about protein DNA binding events right now. And then you have your homeo domain, and these are your homobox proteins, or your homeobox proteins. And these are really interesting because they essentially cause things to be patterned. So here we can see this is a wild-type Drosophila down here that has its 2 wings, 6 legs. They're all coming out of its thorax. If we start messing with these homeobox proteins and start moving them around, then here we have one indication where the legs developed where the antennae should be. Here we have an additional mutant where we formed additional wings. That all comes from messing with these homeobox proteins, which set up the regulation of how do these different organisms pattern. And Drosophila, they do this all the time. So then we have protein-protein interactors. And so those are going to be your leucine zippers shown over here. Remember that your leucines are on this hydrophobic face where the helices are going to interact, and then they have a basic region that binds your DNA down here. And then you also have a helix-loop helix as opposed to the helix-turn helix, which they're all kind of the same. But essentially you have your helix here, your loop, and then these have a DNA binding domain on them as well. They're called protein-protein interactors, though, because the 2 different subunits are both considered proteins. So remember the difference between prokaryotes and eukaryotes in terms of the way that their RNA looks. So here is your DNA. You've got your coding regions that are shown here that give multiple proteins. They're polycistronic, meaning that you have one promoter, and it's going to encode for multiple genes. And then you've got your eukaryotes, which have, again, a 5' non-coding region and a 3' non-coding region, as opposed to that's missing up here. You have a poly A tail, you have a cap structure, and it gives you a single protein. So very, very different. Don't forget the differences between prokaryotes and eukaryotes when you're talking about their structure. So then when we talk about the regulation and how that differs, we just looked at the different motifs. We briefly looked at some of the different regulatory systems. And so here's just our list of the different ways that eukaryotes versus prokaryotes. And actually, that should be prokaryotes and that should be eukaryotes. Sorry, guys. That should be flipped. Because prokaryotes have the operon organization. They use positive regulation. They have this transcript attenuation. So they actually have a system where they can suppress the transcript. And they've got this SOS system, which is basically a stress response system, where they can change one thing and it gives a global regulatory effect. It's called an SOS system. They can repress translation. They can do post-transcriptional regulation. Sometimes you have these sRNAs that act in trans, or you have robo-switches, and then they can do genetic recombination. We can have genetic recombination in eukaryotes. It just doesn't happen as frequently. Prokaryotes do it all the time. So remember, we're talking about bacteria there. And eukaryotes, We have actual structural changes in the chromatin. We can have transcriptional activators and enhancers. We have these very large complexes of proteins. We can have hormones. Remember, we have steroidal hormones and non-steroidal that can interact on the cell surface to initiate cell signaling cascades, or we can actually directly affect that. We have these ncRNA or non-coding RNA. That's very, very important in regulation. The patterning of the homeobox proteins, we see that a lot, and then differentiation from stem cell down to all those different cell lines where we can interact at multiple points in that process for regulation. So that just very quickly covers regulation of gene expression and how they differ between prokaryotes and eukaryotes. Questions? You will see very little of this on your test. You might see something about an operon. That's probably about it. We want to start with A. I think. Yes, we do.
First round, the Jeopardy round.
Okay, so this is a way for you guys to earn a lot of extra credit today. Because essentially what you're going to do is if you answer a question for 100, you will get 1 point. If you answer a question for 200, you'll get 2 points. 300, 3 points, and so forth. You can earn up to 50 points. Okay? So we need to divide into equal teams. So divide yourselves. 2. Yep. Yep, let's do 2 teams. Remember, you're keeping track of your points to turn into me at the end. Do we actually end up with equal teams? Nice. Okay. So remember that you need to rotate who answers. So it can't be the same person answering every time. If you miss the question, don't say anything because the other team will get a chance to answer it for half the points. Okay? All right. You can look stuff up, but there is a time limit. You can't spend like 5 minutes looking up something. Okay? So we're going to start over here. Where do you guys want to start? You can talk as a group. Yes. Okay, we're going to start at the front.
Okay, DNA for 100.
Okay, DNA for 100. Oh, I should tell you, they're in no order of complexity. That's too hard, you guys. Okay, this technique can be used to make many copies of DNA for sequencing or cloning. Replication, good guess, not correct. Replication is a technique, but it's a natural technique. We're talking about a biotechnique. Okay? Okay, that comes over to you guys. Yes, PCR. Okay, where are we going next? Comes over to you guys now. DNA for 500. Okay. These two DNA fragments are produced from the template, and I give you the sequence of the template using dideoxynucleotide method or Sanger sequencing with a DDC. You actually have to tell me what the fragments are. Mm-hmm. So how does Sanger sequencing work? It's asking for the two fragments. And so if I'm going to put in DDC, what fragments am I going to get out if I'm doing a Sanger sequencing method? Do you guys have an answer? Okay, go ahead. Okay. So that's fragment 1. What's fragment 2? Okay. Yes. So nice job. So remember, Sanger sequencing is putting in that nucleotide that's stopping the sequence on that base. So anytime you have to incorporate a C, you're stopping the sequence. And so the DDC is that base. So technically, it is most correct to say GTDDC, indicating that you've got a DDC that's going in there. Questions on that? We got it? Okay. All right, back over here. Amino acids for 500. The amino acids TYR, ASN, and THR have the following common property. What do you guys think? What's common about these three? What? Okay. One thing. One thing that's common. You're right. They are polar. So remember how those amino acids are grouping, okay, guys? So that you can look for commonalities and patterns. All right, back over here. RNA for 500. In the spliceosome, U1SNRNP binds at the 5' splice site, followed by U2SNRNP binding at the blank. Guys, I'll post these up tonight so you don't have to necessarily try to write down everything. You guys have an answer? Yes, the branch site. So remember, U1 is there, U2 comes in on the branch site. All right, back over here. Enzymes for 500. A technique that can be used to study three-dimensional protein structure is this. There's a plant that just fell down out of it is a plant, yes. Do you guys have an answer? Yes, X-ray crystallography. Nice job. Okay, back over here. Yes. Oh, good question. Okay, X-ray crystallography is going to give you three-dimensional structure. NMR a lot of times is giving you two-dimensional structure. You can get some hints at three-dimensional structure if you use the right pulse sequence. But typically we think of NMR as being more secondary and X-ray as being more tertiary. Good question. Okay. The last 500, okay. The concentration of acetic acid in 250 milliliters of 100 millimolar acetate buffer at pH 4.76. There are calculations in here, just so you know. You guys are going to be allowed to have a calculator on the final, but it can't be a programmable one. Okay? Do you have an answer? It's 50 millimolar. You didn't get a bonus. Did you guys have 50 millimolar? So remember that we have, remember that when we go back to, we've got 100 millimolar, and if you have complete dissociation, then it's 50 millimolar of acetic acid, 50 millimolar of base. Okay? All right. No one got that one. Okay, back over here. Which one? Enzymes for 400. This level of protein structure is composed of alpha helices, beta sheets, and turns. Do you guys have an answer? Yes. What is secondary structure? All right, back over here. DNA for 400. Regardless of if they produce blunt or sticky ends, all restriction enzymes have this property in common. Actually, this question should more accurately say, what's the common property of their recognition sites? You guys have an answer? Kind of? Is that what you just said? Okay, 15 more seconds. Oh, nice. Catalytic triad, but no, it's not that. Over to this team. Nope, not quite. I was looking for the fact that they're a palindrome. Remember, they have that palindromic sequence. Okay, back over here. Oh, we already did that one. Sorry. So you have RNA for 400, amino acids for 400, amino acids and pH for 400, or anything less than that, basically. Okay? These are the charged group present in glycine at pH of 7. So what parts of glycine are going to be charged at pH 7? Do you have an answer? Correct. Yep. So typically we talk about the amino group and the carboxylic acid group. So that would have been okay. Exactly what Jacob said was okay. If it's in a long group, like a peptide, then you're talking about the amino terminus and the carboxylic terminus, or the C term, N term. But when you're talking about a single amino acid, usually we don't term them C term, N term. Okay, back over here. RNA for 400. This type of RNA is used by scientists to knock out gene expression. CRISPR is a system that has an enzyme Cas9 that cuts, and then it uses guide RNAs to cut in a specific place. But it's an actual system. Okay, over here. What do you guys think? siRNA. Yep. Okay, back over here. When the pH is more than 2 pH units above the pKa of a carboxyl group, then the acid is blank. There's basically two choices. Is it going to be protonated or deprotonated? Yep. Unprotonated or deprotonated, it's the same thing. Yep, okay, back over on this side. What do you guys think? Enzymes for 300. A protein is considered to be blank when it is converted into a randomly coiled structure without its normal activity. There's a specific term for this. What is? Yeah, denatured. Good. So on one of your tests, you had a question about a prion protein, and it talked about how are you going to determine the structure. Do you guys remember this question? And it said you're going to heat it up and then you're going to use X-ray crystallography, I think, or you're going to heat it up and use NMR. Prion proteins don't denature. That was part of the tricky part of that question. They don't denature at all. So they won't denature under heat. You have to do a lot more rigorous things. But a lot of times you'll want to denature a protein before you run any kind of analysis on it. Okay, back over here. This amino acid has an ionizable R group with a pKa near neutral pH. Do you have an answer? Nope, not lysine. Good guess. Remember, lysine is very, very basic. So its pKa is like 10.92. Okay, over to this team. What do you guys think? Okay, 10 seconds. Okay, which one? No. It is histidine. Remember, histidine has that very strange ring structure, and its pKa is so near neutral that it kind of has that weird, it can either be protonated or deprotonated, which is why we find it in a catalytic triad all the time. Okay? All right, now I messed up. We're over here. Okay. Okay, where do you guys want to go? Okay, DNA for 300. These 3 genetic elements are necessary when using a plasmid for cloning. You guys have an answer? It's 3 things that are necessary. Seconds. Ooh, really close. You got 2 out of the 3. Okay. Over to this side. What did they get? Nope. Nice try, though. Okay, 10 seconds. Okay, what's your answer? You have the same answer. Okay. So it's an antibiotic resistance gene, an origin of replication, and a promoter site. You have to have a promoter site for RNA polymerase to work. So okay, over here.
Answer, Daily Double. You and Spencer have.
So how many points do you guys have? So you can risk up to 5. You can risk up to 5 points. That's it. What's the enzyme? You guys have a guess? Ooh, no, close. It's not it. You guys can answer it, but you only get 1.5 points. Because it was a 300-point question. So you don't get their daily double. So what enzyme is it? Seconds. What? No, not a restriction enzyme. It's a specific enzyme that always makes me think of tomatoes.
Dicer.
It's dicer. Yeah, it's dicer. Dicer is the name of the enzyme that goes in and cuts long RNA into single units. Or into small little chunks. I shouldn't say single units. Into small chunks. Okay, over here. Hydrophobic molecules are driven together by blank, not because they have an affinity for each other. You guys have a guess? Yes, entropy. All right, over to this side. Which one? Enzymes for 200. The configuration of most peptide bonds in a protein is blank. You have two choices here, cis or trans. Do you have an answer? Yes. It's trans. Okay, over on this side. Amino acids and pH for 200. The distance when two atoms no longer repulse each other, yet have the strongest attraction is known as the blank contact distance. It's also the name of a bond. You guys have an answer? Yes, van der Waals. All right, back over here. Which one? Amino acids for 200. This amino acid forms disulfide bonds. Which one? Yes, cysteine, because it has that sulfur moiety in it. Okay, back over to this side. RNA for 200. This is typically not found in mature mRNA molecule. Don't get too specific on me. You have an answer? Introns, good. All right, back over here. This 6-nucleotide primer set would produce the following PCR fragment. So this is the PCR fragment that you get out of having these two 6-nucleotide primers. What are those two primers? Ready to give it a shot? Okay, 15 seconds. Okay. All right, other team? What do we think? Yes. Yes. So remember, primers have to be on the ends because then they're going to amplify what's in the middle. There's two of them. In order for PCR to work, you have to have two primers. You have to have a forward and a reverse. Okay, which one? Where are you starting? The 3' end or the 5' end? Okay. So what is it? Well, so yeah, because remember it's the match that produces it. And we're going to report them 5' to 3'. Mm-hmm. Yes. Yep. So the only thing you forgot to do was flip that one that you had on the 3' end, because they report 5' to 3'. So if you're given a fragment and you're asked for what primers produce the fragment, all you have to do is look at the ends, and then you have to make sure you have the complement of those ends, and then you flip whichever one. You start at 3', you flip it so it's 5', 3'. Make sense? Good. All right. Now let's see, I'm over here. Okay, where do you guys want to go now? Enzymes for 100. The term quaternary with respect to protein structure means this. Do you have an answer? Yes, it has multiple subunits that are coming together. Good job, guys. Okay, over here. This term describes a molecule that contains both positive and negative charges, but overall has a neutral charge. Remember, it's that German word. Yep, a zwitterion. back over here. Electrostatic interactions between atoms with opposite electrical charges are also called this. No, not van der Waals. Ionic, yes. Or they're also called salt bridges. Okay, last question, RNA, over on this side. This nucleotide is used in place of T in RNA. Yep, just you. All right, so I have another one, if this was helpful, and we can do another one, but let's take a break, and I'll pull up the other one, okay? I don't know why not.
First round, the Jeopardy round.
Oh man, bad stuff went down out here with plants. No, they're just really prickly. I feel bad that their plants fell over. Oh, I'm sure he's fine. He always has a million things going on. So. Yeah, I'm sure it's just fine. Okay, now I've stabbed my hand sufficiently with plants, so you guys ready to start? Okay, we started on this side last time, right? So we're going to start over here this time. So what do you guys think? Enzymes for 500. This model of allosteric behavior, also called the symmetry model, proposes that allosteric proteins have two distinct states designated as R and T. So I should tell you this is called B because these questions are harder in general. What the name of the model is? And this time we're not going to go back and forth. It's just single team gets an answer. There's only one that I can think of, but yeah, if you find multiple answers. Okay, do you have an answer? It's the Monod-Weinheim-Shangu model.
I didn't think that was going to be right. Yeah, I have it.
It's the name of the people that came up with this model. Remember that your R state is relaxed and T is tense, and so that's like an open and closed conformation. And the name of the model is actually named after the people that did it. Do I know what it is? Yes. So that is essentially, yeah. Yeah, it's essentially the same. Well, I'm going to give you half credit for it because you didn't tell me the name. And the model is actually named this. Okay? All right. Okay, over on this side. Amino acids for 500. This amino acid provides nitrogen or ammonia for many metabolic reactions that use nitrogen as a substrate. Glutamine. Yeah, I'll give you guys. You can get half credit for that one. So glutamine. Okay, back over here. Oh, potpourri just means it can be anything. For 500? Okay. Aspirin binds and blocks this enzyme, thus explaining its multiple effects. One or two? Actually, it doesn't matter. So I will give you credit for it. This question actually says prostaglandin synthetase, or synthase, but cyclooxygenase it also binds. So I'll give you guys credit for that. Yep. Okay, back over here. Kinetic perfection is when this ratio, oh, that should be ratio, not ration, is at its upper limit. So what ratio is at its upper limit when we reach kinetic perfection? Yep. Your KCAT to KM ratio. Oops. Why did this not? Okay, it's going to go through everything again. Sorry, guys.
First round, the Jeopardy round.
Up as all of our 500s being blocked out. Okay. Yeah. Okay. pH and thermodynamics. In the absence of an enzyme, when the energy of activation is high, it may be blank to an exergonic reaction to proceed. Do you have an answer? It's not really technical. Ten seconds. Okay, what's your answer? You have the concept. You have the concept. It's called coupling. So remember, a reaction that produces energy is coupled to a reaction that uses energy. So you have the concept. No. No point. Okay, back over on this side. Enzymes for 400. Which one of these is the most potent inhibitor? I'm just looking for a letter. No. It's B, because the smallest Ki is the most potent inhibitor. What you were looking at is the rate of reaction versus the substrate concentration. And inhibition, because of the different types of inhibition, can fall anywhere in that and doesn't necessarily indicate which one is most potent. So then you need to look at the Ki, or the kinetics of inhibition. Okay? All right, back over here. Who's choosing? Okay, potpourri for 400. Glutamate is the precursor for the amino acids glutamine, proline, and blank. Do you have a guess? Yes. Arginine. I don't know why it came up with 100 at the bottom. Don't worry about it. I don't know why. Okay. Yeah. Something's wrong with the game. There is no 400. Disregard the 400 in potpourri. Okay. Over here. Which one? Amino acids for 400. For the peptide GPGSQQ, the expected charge at pH 4. What is the expected charge at pH 4 for that peptide? Do you guys have an answer? Negative what? Plus 1. So remember, guys, remember, don't forget your ends, right? So you've got your carboxy terminus and you've got your amino terminus, and you have to make sure whether or not those have positive or negative charges, regardless of what the amino acid is on your C term and your N term. And then you add them all up, okay? All right, back over here. Given that the pKa values for phosphoric acid are 2.14, 6.86, and 12.4, this is the ionic form that predominates at pH 4. So you're looking for a formula here. So remember, you've got your formula, and then depending on where it falls, you have either, a couple of minus or a couple negative charges, or you have a couple positive charges. So the question is how protonated or deprotonated it is. So what's the charge on that? Okay, what's the formula? Oh, not a mathematical formula, a chemical formula. HPO. Yes, H2PO4 negative. So remember, where it falls in that spectrum determines how many negative charges it has, because it looks at how protonated or deprotonated that's going to be based on the pKa. So yes, it has one negative charge. Okay, back over here. Okay. If the enzyme concentration is 5 nanomolars, the substrate concentration is 5 millimolars, and the Km is 5 millimolars, then this is true about the relationship between the enzyme and the substrate. So what can be said about the enzyme and the substrate? Do you have a guess? Okay, 10 seconds. Okay, go ahead. Yes, it's saturated. How did you know that? The enzyme is saturated with substrate. How do we know that that enzyme is saturated with substrate? What made you guys think that? Lucky guess? Nope, they weren't. They weren't all the same. So the enzyme concentration was less than the substrate concentration. So when your substrate concentration is really, really high, then every enzyme site is bound up by substrate. So you reach a saturation. All right, back over here. Which one? Potpourri? The electrons for the reduction of molecular nitrogen are donated by this molecule. You guys have a guess? it's ferredoxin.
Last time we didn't go there, that's really cute.
Okay, back over on this side. Enzymes for 300. The slope of a Lineweaver-Burke plot. It's looking for an equation. You just really got into ferredoxin, huh? Do you have a guess? Km over Vmax. Good job. All right, back over here. Which one? Amino acids for 300. Substituting asparagine for glutamine is considered this type of substitution. What's your answer? No, you're not. I think this is the wrong answer. So, no, it should be non-conservative. Anytime you're changing charge, it's non-conservative. Yeah, I mean, yes, it's non-conservative. Sorry, I made a mistake here. Yeah, you gotta keep me honest here. Okay, over here. Enzyme kinetics.
Answer, daily double. You and Spencer have been seesawing back.
Okay. So it's timed, and you can risk up to 5 points. Okay, how many are you risking? 2. Okay. Value is equal to the substrate concentration when the reaction rate is half of its maximum. Okay. What's your answer? Yep, KM. Okay, back over here. Given the amino acid sequence lysine, phenylalanine, leucine, asparagine, and methionine, the alpha helical structure formed at pH 5 is stabilized by this type of interaction. Okay, what's your answer? What do you mean a second structure? Yeah, so that's the secondary structure. So in this particular one, it's really hard to go backwards on this. So on this particular one, because of the charges, you're going to have some ionic interactions. Well, alpha helices are typically going to be hydrogen bonds. Again.
First round, the Jeopardy round.
And then it's just going to randomly put them up here. All right, back over here. Which one? Enzymes for 200. Km is the blank concentration that yields half the Vmax. What's the answer? Yep. Okay, back over here. Metabolic pathways that have alternate products are often regulated by blank and blank. Do you have an answer? Close. Inhibition and activation, which is kind of negative and positive, but the words are important here. Okay, back over here. In a catalytic triad, this amino acid is often the nucleophile. Do you have an answer? Yes. Good job. Okay, back over here. This one? pH and thermodynamics. Oh, man. See, this is the problem with this game. As soon as I mess up, push the wrong thing.
First round, the Jeopardy round.
Okay, pH and thermodynamics, right? When preparing 50 milliliters of a buffer with a pH of 4.5 using CH3COOH, which has a pKa of 4.74, and NaOH, use this amount of base. Yes, 0.575 molar. Okay, back over here. This term is used to describe the dynamic recognition of substrate when binding to an enzyme occurs. Okay, what do you guys think? Induced fit. Good job. Okay, back over on this side. Glutamate is the precursor for the amino acid. Didn't we already do this one? We already did this one. Okay. This enzyme catalyzes the committed step of fatty acid synthesis. You only get 100 for it, and actually you only get 50 for it because you didn't say carboxylase. Nope, you were done. Okay. Back over here. An enzyme changes the reaction blank, but not the equilibrium. Yes. Okay, back over on this side. This amino acid is most likely to be found in a zipper. Yep, leucine. Okay, back over here. For an enzyme that follows simple Michaelis-Menten kinetics, what is the value of Vmax if V sub naught equals 1 micromole per minute at 10 km? Oh, close. 11 micromolar per minute. I'm not sure where you got 1.1. Yeah, okay. Yeah. So this is micromole per minute. I'm not sure again, exactly where that factor of 10 came from. But remember, for this equation, you're going to assume that the substrate's very large, so then you know what the max is. Okay? All right, pH and thermodynamics. Last one. Below its pKa, a molecule is in this state. Yep. Okay. So make sure you turn in your sheets of paper. Show me how much extra credit I should add. Keep reviewing old lectures. Remember that it's really heavily reliant on glycolysis, gluconeogenesis. Those are going to be heavily weighted typically on the test. They really love that stuff. You can do it one per group, that's fine. Tomorrow you're going to do immune system. Then I'll be back tomorrow. I think that's it. What? Probably not. All right. I will definitely look through it. Can't guarantee anything about throwing out questions, but I'll definitely look through it. Okay? All right. Thanks, guys. Have a good rest of your night.